thank you, Steve, Joe, and Sandy, for all the things you're doing with CZ BioHub. And it's nice to be able to zoom in here to your 5th year celebration and say a few things from my perspective as NIH director, and particularly to say how exciting it is to see the kind of amazing critical mass of talent, expertise, technology, vision that the BioHub has created that you're all celebrating here today. I got asked to say a little bit about partnerships because I think that's a really important thing for us all to be focused on. So I will. I might also say a little bit about COVID-19 because it's a necessary obsession of myself and many of you as well. And a little bit about how important it is that we're investing in our most important resource, which is the people to do the work, especially the next generation. So I'm going to share my screen, assuming that this goes the way I want it to, and we'll see whether you can see it when I go to full screen. Are we good?
Looks great.
Okay, so yes, innovative model for biomedical research, and you're doing a lot of innovative modeling yourself. And of course, I have to just gloat for 1 minute here because it is October 4th, 2021. And NIH likes to believe that we do a pretty good job in funding exciting research. So here we are on this very day with the Nobel Prize to these 2 investigators at UCSF and at Scripps for their remarkable work finding discovering receptors for temperature and touch. And yes, they were both supported by NIH grants for more than 20 years, and we're very proud of them, and they continue to do great work. I think that gets us up to 165 researchers that have become Nobel laureates. And I would predict some of the people listening to this right now will be on that list at some point in the future. And if so, we hope we supported you and did the right thing to help you. Okay, so I'm going to talk about accelerating biomedical research, and I'm going to have 3 areas to talk about. First, partnerships, then COVID, and then the next generation. And all of that in supposedly 15 minutes. We'll see how I do. So partnerships make everything go faster. Well, yeah, we are NIH. We are the largest supporter of biomedical research in the world. But we basically, while we run an intramural program, which is pretty amazing in terms of the science that's going on there, most of our funds go out there to academics who do great work, as you all are doing, and make discoveries that then lead forward to advances that ultimately affect the clinic. And so industry is necessary there in order to make this as successful as possible. And one of my goals during my time as NIH director is to be sure we are taking full advantage of those industry partnerships like the Accelerating Medicines Partnership, or something I'll tell you about in a minute called ACTIVE. Philanthropy is critical in there as well, oftentimes focused in a particular way on a particular area. I'm delighted that the way that CZI has undertaken this is very much focused on basic science because that is an area of exceptional opportunity. And of course, none of this makes sense if we're talking about trying to take things to the clinic without our most important participants, which are the research participants. So that's our ecosystem. And it's certainly, I think, in all of our best interests to be sure we're actually nurturing that. So I'll give you a couple of examples that came to mind of these kinds of partnerships. One in the basic science arena, very familiar to this group because CZI has been a big part of it, is the effort to build a framework for molecular mapping of the human body at single cell resolution, even with some degree of spatial transcriptomics now included in it. So HubMap, which is collaborating with lots of other efforts in this way, is moving forward to map lots of human organs. Here's an example of some of the data in HubMap. This happens to be human spleen with a nice UMAP description or display of the cell types that are present there. And of course, this is a highly collaborative effort with multiple partners. And I do have to mention, of course, CZI as one of them. That's a good example. I'll give you another example of a partnership that's really very different and very out there in terms of its boldness. This is an effort we started 3 years ago with the Bill and Melinda Gates Foundation to try to take the things that we are learning about gene delivery, gene editing, and in vivo approaches to do 2 outrageous things, to basically come up with a way to cure HIV where you don't need ongoing therapy anymore, And while we're at it, let's cure sickle cell disease. And let's do this in a fashion that would actually work in a low resource setting, basically requiring you to develop an in vivo delivery of a gene editor, which on the one hand could go and find the proviruses that are lurking in the system of people who have HIV, which means figuring out where they are. And for sickle, would home to the bone marrow, and either by turning on fetal hemoglobin, by activating that system, probably by targeting BCL11A, or maybe even by making a specific fix of the sickle mutation. This is a 10-year program, but if you don't set those kinds of very bold goals, they're not going to happen. And there are some overlaps here between HIV and sickle, in case you're wondering, how did these two get together? Well, you can see what those are. And so we are doing this jointly with Gates and NIH and making some pretty good progress, although I will say this is truly a reach to do this in 10 years, but we're determined to do it as much as we can to get there. Okay, second topic is COVID, and you've no doubt been talking about that because we all are talking about that, whether it's at the coffee shop or the bar or in the lab, because this has been the greatest challenge for science in our generation. And I gotta say, science has risen to that challenge in remarkable ways. We all kind of remember when it looked like things were getting serious here, early January 2020. I certainly remember that, sitting down with Tony Fauci at about that time, January 9th or 10th, when the sequence of the virus was posted on the internet. It was clearly a coronavirus, and it was time to start the process of building vaccines. And as everybody knows, there are multiple different platforms that were put into place here and funded rather significantly by the US government to speed up the process of developing the trials and also to manufacture in advance in case any of these worked, sort of at-risk manufacturing. The ones that are furthest along and got there quickest are based upon mRNA, this remarkable new development, which makes it possible, once you know the sequence of the pathogen, to develop the vaccine or design the vaccine in a day or two, which is exactly what we did at NIH working with Moderna, and which 63 days later was starting a phase 1 trial in volunteers. And 11 months later was approved by FDA for emergency use, both the Pfizer-BioNTech version and the Moderna version. And of course, other vectors as well, adenovirus or just recombinant protein, are coming along, and J&J is also, of course, as you know, approved in the US. And yes, I don't think when we were starting down this road, I would have dared to hope for efficacy of 94 to 95%, but that's what we got. Again, remarkable efficacy against symptomatic disease. Now, a lot has happened since then in terms of the arrival of various variants like Delta, and the fact that we've learned that vaccine immunity wanes over time, most effectively demonstrated in Israel with data they've generated, which is really quite elegant indeed, and which is now the motivation to come up with a third dose, a booster, which does seem to be capable, again, looking at Israeli data, in greatly reducing the incidence of infections, certainly severe infections, which is what we're most worried about. So all of that happening. Meanwhile, what about therapeutics? In March of 2020, when I looked around to see what was happening with therapeutics, much of it supported in academic institutions, frankly, it was kind of a scattershot. There was no real coordinated effort to try to prioritize the various therapies that had been suggested. And many of the trials that were being run were on hydroxychloroquine, and many of them were small, underpowered, and unlikely to give a compelling result. So we pulled together academics, NIH, and industry, FDA and CDC joined up as well, for something called ACTIVE, Accelerating COVID-19 Therapeutic Interventions and Vaccines. Yes, it had a vaccines part as well. The reason the vaccine trials all followed the same master protocol of 30,000 participants with the same endpoints was because ACTIVE came up with that as what they felt would have to be applied for a trial that really could be trusted to give a reliable result. So this group, in the space of just about a month, came together on April 17th and has been in continuous operation since then, with about 100 people who dropped everything. And that includes people from industry, academia, and NIH, breaking themselves up into 4 different working groups, designing master protocols, prioritizing therapeutic agents, coming up with ways to organize clinical trials in a way that has never been done before, where you pulled trial networks together that hadn't really worked in that way in order to speed up the process of testing out various therapeutics. And this has now tested more than 20 different therapeutics, most of those being repurposed, hoping that there would be something there in the existing set of drugs that had already been in humans that would turn out to work. And we got remdesivir out of that, and we got monoclonal antibodies that were developed specifically for this. But frankly, most of the repurposing efforts did not yield a successful result, although there's still some going on right now, and this is far from over. The stakeholders in this was truly a dramatic group of people who agreed to work together in an unprecedented way. 20 companies, pretty much all the ones that had developed vaccines and therapeutics, a whole bunch of government agencies, nonprofits, and the foundation for NIH basically becoming the program managers, and very ably so, to keep the science going forward in an incredibly efficient way, running something like 40 to 50 meetings every week of groups, working groups and subgroups to push this agenda along. What did we learn? Well, I think in a general sense, and there's a whole lot of data underneath this about the 20 compounds that have been tested or are being tested now, is basically if you think about the course of illness from somebody who has asymptomatic disease, going on to mild symptoms, going down to get more sick and ending up in the hospital with no oxygen, with oxygen, high flow oxygen, and ultimately ventilation. Antiviral strategies clearly are most successful if applied early. Now, we have seen that with monoclonal antibodies. They don't work in hospitalized patients, which was a big disappointment, but it is the answer. Immunomodulatory strategies like dexamethasone, on the other hand, work in people who are really sick in the hospital, because at that point, it's pretty hard to find the virus anymore. It's the immune system that is overreacted to the threat and is doing more harm than good. And hence, you need something targeted to try to turn back that dial. Interestingly, anticoagulation strategies have turned out also to be a valuable assay, but you got to be careful about how you apply them. This is a virus that likes to induce clotting, both of small and large vessels. And so full strength heparinization turns out to be highly beneficial early in hospitalization, but not later in hospitalization, where there is no benefit and there might even be harm. All of those subtleties had to be worked out by organizing and designing trials in a fashion where you could really believe the results. So my hat is off, speaking of a partnership, to the people that made this possible and continue to do so even today. But of course, what we really wanted was that remarkable antiviral oral agent that you could take as soon as you got a positive COVID-19 test, and it would knock the virus out and you'd never even have to do anything more than that. And we hoped there might be one of those in our repurposed collection, but frankly, it didn't turn out to be as exciting as we had hoped. We got some singles and doubles, to use a baseball analogy, but we didn't get home runs. But it's time to do that. So beginning now several months ago, this APP, Antiviral Program for Pandemics, with $3.2 billion from the Congress, aims now to try to look in a very drug design approach. How could you put together small molecules that block the viral life cycle in a way that would be very specific and should be low in toxicity, because they're blocking the virus. And the virus has a protease that's a particularly attractive target here, but also an RNA-dependent RNA polymerase, which could be a good place to go, since human cells generally don't have that. There are 3 or 4 compounds already being pushed through in this space, some inactive, some by companies on their own. And the one that just got a big splash this week is the one which was originally developed at Emory, again, I have to say, with an NIH grant, originally called EIDD2801, and is now this announcement that you saw just in the last. This is from April 16th, where the phase 2 trial is being talked about, but molnupiravir just a couple of days ago, cutting hospitalization risk in half in a clinical trial of 775 people, and with a very encouraging safety profile, really no significant adverse events. And all this data now quickly going to go to FDA to see if they would approve it. If this is something which FDA finds to be approvable, that really changes the dynamic where you have the opportunity of an oral agent that can be taken, it's twice a day for 5 days, which would, in many instances, especially with high-risk people, reduce the terrible carnage that this virus has caused. Now, 700,000 people dead in the United States alone, 5 million in the world. Maybe this is a moment. Now, again, I don't want to jump the gun. FDA hasn't actually reviewed this yet. Obviously, there's no publication. It's a press release. So one needs to be careful about assuming what this means, but it certainly is a step in the right direction. And the APP aims to take this further with multiple other antivirals, because there's a good chance you will want combinations here in order to avoid the emergence of resistance. I also want to tell you about our diagnostic effort, because I know that the BioHub has gotten engaged in this in a very significant way by providing COVID-19 testing in California. Back in April of 2020, as we looked at the situation with testing, it was pretty clear we had a lot of work to do to try to expand the capacity for doing testing, and also to move more of it, if we could, into point of care instead of requiring a centralized laboratory with all of the delays that can be associated with that. The Congress gave NIH about $1 billion on April 24th of 2020. 5 days later, we started this program called RADx. It was basically an intent to accelerate innovation by encouraging people who had great technology ideas to come forward and let us look at them. And if they looked promising, put them into what we called a Shark Tank. And basically, this was an innovation funnel. This was NIH acting like a venture capitalist, but with a remarkable team of technologists, engineers, business experts, people who understood supply chains and manufacturing, aiding in the selection about which of these ought to go forward and get to the final point of being out there in the clinical application. We got 3,000 applications, most of those in the first couple of months. And then those were sifted through. 824 of those got complete applications. 179 of those went into this first phase, which we did call the Shark Tank. And that was an intense week where the experts were looking at the technology, trying to figure out what its strengths and weaknesses were. If it looked good, the applicants then got some money to go to phase 1, where we helped them get validation and actually to get FDA emergency use approval. And then phase 2, if things still looked good, to do clinical tests and scaling up. And there are now 35 of these platforms that are out there in clinical use. And that includes a whole lot of point of care and over-the-counter measures, which has been a big step forward. So if you look in your local pharmacy and you see tests that you can take home, one of them is the Quidel QuickView. That was a RADx product. Another one is Illum. That was a RADx product. So this has been quite the experience. And it excited me by showing that we could do things scientifically in a public health crisis in a very different way, a very nimble way, a risk-taking way, to be sure, but it really worked. So lessons learned, we're already all thinking about that. I bet you are too. We are not seeing the last pandemic. This one is far from over, of course, but there will be something else out there. We already need to be thinking about those lessons and starting the process of figuring out, if we know what the most likely next pathogens are going to be, and people are pretty good at putting together a list, about 20, shouldn't we start right now to develop the vaccines that would be necessary and get them all the way to phase 1 trials? No reason not to do that if we have the funds. Therapeutics, should we look at all of those pathogens and figure out what are their most vulnerable targets that we can start to develop small molecules using a lot of the more powerful tools that are now out there to do so? And with tests, should we take what we've learned with COVID-19, because many of these platforms are very generalizable, and actually begin to imagine how they would be applied if the next pathogen was influenza, or maybe it was a filovirus? Are we ready to do that? I wrote a little piece in Science about this, but there's much more that could be said. What we must not do here is to step back into complacency, which unfortunately has always been the case in the past. But now, after going through, and we're not even through, the worst pandemic in more than 100 years, it has now taken more lives in the United States than influenza did in 1918-19. we really have to be dedicated to pursuing the next steps and not letting that opportunity go by. And I think there's a fair amount of support for that, both scientifically and in the Congress, but until we actually have it in place, I'll be a little worried that other things might start to occupy people's minds. Well, final thing I wanted to say is all of this is great, but speaking to the BioHub, with all of the talent that you all are investing in, the future really depends on that we need the next generation of scientists. And particularly those who are early in their careers, the early stage investigators. We at NIH have worried about this in terms of the difficulties those folks have been facing over the course of many years, especially as we have seen budgets go up and down and oftentimes success rates tracking likewise. And when we look at the circumstances for early stage investigators a few years ago, it looked like something needed to be done. So this is the number of early stage investigators that got their first grant as an R01 over the course of time from the mid-1990s up until now. And you'll see things were around 1,000, which is sort of healthy, although it should be higher. And then, despite the fact that we went through this doubling of the NIH budget here, a lot of the money was going to other people. A lot of them were people who had a lot of funding already. And then as the budget flattened after the doubling in 2003, what was going to early stage investigators went down. Now we did a little correction here, giving early stage investigators a boost in their priority scores, got things a little better, but then we had the sequestration, this awful experience back in 2013, and took a nosedive. Well, what's happened since? We have totally made this a high priority, the highest priority, that if you're an early stage investigator and you get a grant that is funded, is recommended for funding in the top 25%, you ought to get paid. Even if everybody else is at the 18th percentile, you should get your dollars. And that's what we have been asking the institutes to do, and look and see what's happened here with that. So this just goes to 2020. I will tell you 2021 is off the graph here. We just got those numbers, 1,386, sorry, 1,486. So we're setting a new record here. And that is, I think, exactly what we should be doing to try to nurture and encourage the talents of all of these up-and-comers who are going to be our future. And just finally, I would say we have other programs like that. I guess I'm doing a little advertising here for people who are listening. If you have a great idea that's a bit risky and you're a bit concerned that a standard study section might sort of look at it and go, oh, I don't know, maybe you better have more preliminary data. We want to encourage those kinds of risk-taking investigations. And so that's why we have things like the new Innovator Award, early stage investigators, single PI, not expecting to have a lot of preliminary data, but a big, bold idea. And similarly, I'm concerned about how long it takes to get our trainees to the point of being independent, which is why I started this particular program, the Early Independence Award. And actually, I kind of learned a bit about this from people like Joe DeRisi. And this is, I think, turned out to be one of the most exciting programs. We don't fund very many, maybe only 12 or 15 every year, but these are the most amazing applicants because they're ready to become independent after their graduate training. And they have bold ideas and they have been remarkably successful. And these have been around long enough now that we can start to evaluate productivity. And they look extremely good, better, in fact, than a typical R01. So I want to throw all those ideas at you. It's a bit of a smorgasbord, but maybe somehow that fits somehow to what you're talking about today with the BioHub. And with that, I want to thank you for the chance to come and land in the middle of your exciting day and say congratulations to the BioHub, to the Chan Zuckerberg Institute for having the vision to create it. And wishing you all of the best for your next 5, 10, 50, oh heck, 50 years, because we got a lot of work to do. And it's the best time imaginable to be working in science right now because so many things are possible that just weren't even thinkable a decade ago. So I'll stop there and say thank you very much.